So yeah, it's a pleasure actually for me to introduce you to the job profile. What I'm here, especially that I also know the other side from the academics quite well, as you heard that I spent quite some time, and you will hear that later in my presentation as well. Quite some time in academics, and now I might have a good overview of the advantages, disadvantages of both sides, either academia or pharma industry / industry alone. So Connie, if we can go to the next slide, please. I want to start with a very personal introduction, since as Cornelia already mentioned, we have cultural aspects very high in our spirit. So we really want to know who we are here in Novartis, and therefore it's also important what you do offside of working. And so actually I was raised on a soccer yard, what then became the skating yard in winter, in a very small village in Upper Styria. Sport games of any kind was my company in my youth. But with age and maybe loss of condition, I rather went from sporty games towards hiking. And also when the weather is not that nice, I like to be at home playing keyboard, relaxing. So it's kind of a nice composition here. And as you see in the back in the header, this is actually a picture what I took by myself from the Wilde Kaiser in the Kufstein area. So when you want to get some energy back and you like hiking and being in the nature, I think there is no word to say more to this picture here. Besides my activities, I would define myself as being curious, and with curiosity also ambitiousness comes. But during the time getting a bit older, I also learned to be very focused, because you can get lost in details. So focusing and getting resilient towards some stressy timelines is one of my personality, I would say. And also important is my motto, and also my PhD thesis actually was titled in that way, keeping the balance. So you really have to find your balance by yourself. There is no general rule, no general guidance, or no SOP as we call it here, but you really have to find what is the balance for you to really be good in what you're doing and what you're wanting. So when we go to the next slide, please, I want to briefly show you my career path, what is basically academics. I did my master's in Graz in 2008, followed by a doctorate, as Vanessa already mentioned, what I finished in 2012, where I had also some nice time in Cornell University in Ithaca. So a great place to go. Also, nature is beautiful there. I finished then some papers, so I had 2 postdoc years still in Graz, working on basically yeast, Saccharomyces cerevisiae, not for brewing beers, but really as a model organism for doing lipidomics on lean and obese yeast strains to find out molecular pathways, how lipid pathways or lipid metabolic ways in the body, so in the yeast cell, are regulated, and if this is translatable also to higher organisms. After that part, I was moving to Frankfurt, so I did some years abroad. 3.5 years I was at the Goethe University. There I moved a bit from the molecular biology part. So I moved to Frankfurt because I want to see more. I want to broaden my horizon, and there I moved from the molecular biologics towards the biochemistry field, where I did some protein expressions in E. coli and yeast strains, afterwards purification. And so since my goal there was membrane protein interactions towards liposomes, I also generated liposomes by myself and did some cryo-EM studies on them. So I got really kind of a broad background in everything, what means that I'm not expert in anything, but in everything, but I know quite some techniques during that. And after this Erwin Schrödinger stipendium from the FWF, I went back to Graz and wanted to get into a standalone career, working then on lipidomics, but not on yeast anymore, but one stage higher in evolution, let's say, doing flight genetics in Drosophila embryos, and also during aging of this little small nasty flies if you have them at home. But in the lab, they are great. They have a generation cycle of 10 days, and they can get to 70 days old if for your interest. I didn't know that. I was very amazed by the age of such flies. They do have neuron system. They do have insulin signaling pathways. They do have immune systems. So it is really a nice model organism to work with. And the genetics is established since over 100 years. So it was really a pleasure to work there. However, maybe some of you already experienced competition or having a standalone career path in academia doesn't get better these days compared to the past. So I was also searching for new job challenges, not only in academia, but also in industry. So I screened, of course, Austria. There was Viennese industries, of course. There were other industries Europe-wide. I screened for them. However, I was actually not aware that we have such big pharma company in Tyrol. I was not aware of that. Coming from Graz, it's not that far, it's 350 kilometers, but to me it was pretty surprising to have such a big company. What actually started as a brewing, from a historical point of view, it was a brewery. What then I think in the Second World War become penicillin production to kind of heal the wounds or to help the soldiers which got wounded, and then this was increasing, and now I'm here. So my attitude to yeast and beer might be also related to that, and now I'm working in a very beautiful area, but as a principal scientist, as mentioned before, as in the analytic team chromatography, focusing on HPLC systems and driving the projects. So can we go to the next slide, please? So then I came basically really interested in this area here, and by looking at different job profiles, it was not only the analytical scientist profile what kind of tackled my attention, because I've said before, I did some protein expressions, I did chromatographic steps, I did mainly things. So I think I could have applied to at least 5 job positions there. But since I always wanted to be coming from the chemistry side and analytical scientist, I was mainly focusing on being an analytical scientist 100% and not only part-time as it was in academia. And this job description here, where I pointed out the major points, what gained my attention was that you really design, plan, perform and document scientific experiments in a timely delivery manner. So we have to be fast. This is what I heard then in my interview, what I had for the drug products and all that. We have to analyze and interpret, of course, in the early stages of some projects, all the data what we gain and kind of draw our experimental conclusions out of it. This is also a highly team-driven event, what we are doing. So we'll come to that later. So we are not isolated here and sitting at the computer and crying on the screen. We really work as a team and try to help each other and drive the projects further. Training and mentoring. I come from academia. I mentioned this was my daily bread there, and I love to share knowledge and learn from other people. And this only happens when you interact with people. And therefore, this is very important, especially when you work in very controlled. We call it here GXP, so good laboratory practice or other practices environment, where we really have to be very clear in what you're doing with regard to patient safety, what in the end is the utmost goal, what we have always to maintain. What is nice is, and this was something what I was a bit concerned by moving from academia to pharma industry, is that here you also can lead initiatives. So it's not that you stick to protocols. We always have to learn new. We don't have to invent the wheel new, but we have to drive our processes faster. We have to improve quality. We have to reduce the manufacturing steps, what of course improve the quality of the product, because you kind of minimize errors what can happen. And so this is also very well appreciated here, that your ideas are really heard and evaluated. Of course, not all of them are implemented, but they really come to be visible. And this is really something where you get honored in a way that your contribution is really highly appreciated here. So on the next slide, what I put together is my daily work routine. So from the overall packages, what I told you now, we do not have a daily work routine actually here in the analytical department, because in development it's basically driven by the status quo of a project. So if the project is fully developed, you might have more work routine because you have your different methods, your different procedures, what you apply to test for certain products. But in the development phase, where I actually work the most time, you really have to be curious, to be fast, to be smart, to really have this to get the things done which are needed, and not on a daily routine, but you get 2 weeks maybe to develop a method, and then it should be working, as an example maybe. So this is, you have to develop, to optimize, to run, and of course to validate a method, that it's not only working for 2 weeks, but hopefully very robustly over years, to really control the products what we develop. A little bit more than compared to academia, I would say, is the documentation. Again, with regard to patient safety, you really have to document what you're doing in a, I would say, more proper way than in academics. So here we really need to check everything, to document it down, what of course facilitates then troubleshooting what we are doing, but what takes some time. And this is what you have to live with some lab work, what you do besides the fun part. But if we just go to the next slide, I tried to roughly estimate the amount what you kind of do in this particular job as analytical scientist. You spend 15% of your work day in trainings, which is very important to be always up to date, in some meetings which are important, and in also interchangeable in interactions with other teams. 25%, so a quarter, I would say, really goes into documentation at the site, and you have to do this simultaneously to the lab work. But still 60% remain for the fun part, and this is really to be curious, to get things better, to optimize methods, to generate robustness and not fancy stuff. So we prefer here to go the robust way. We don't need fancy methods, we need robust methods. But this is also the beauty of science, to develop something what is robust and works for years. What I forgot to mention is on the right-hand side, you see here two pictures of our machines actually. So the upper one is a solution apparatus, where you can dissolve different products like tablets. So it would mimic, the solution mimics the digestion of a tablet and the distribution of the active pharmaceutical ingredient, the API. And it's coupled to an online UV system, but you also, for more details, you can couple it to an HPLC system and analyze also impurities, which is important to control. What brings me to the lower picture, this is our beautiful new labs with the view on the church in Kundl here. Different HPLC systems, and we have in total 30, I think, in our department. And what people who worked with HPLC systems might appreciate is that they stand on a very low level, so it's pretty easy to exchange the effluents on top of the machines. And this is also linked to an automatic waste system, so you cannot spill the floor. So it really makes the 60% fun part even more fun than it is, as it is. On the next slide, I do have basically my last slide, not to run over time. You might be really interesting why I choose Sandoz here, and what I like really at this job position, having also the experience in academic years. It's here actually, it's basically related to academics. We have here the teamwork. You really interact, you exchange yourself. We have our coffee breaks where we talk about it. We have our Mensa breaks. It's a very good Mensa what we have here, where you just by chance hit some people and ask them, hey, can we do this? Can we do that? This is for method development very important. You don't need to be expert, but you have to know the experts here. And we have quite some experts at this site, I can tell, after being a year in Novartis. However, in decision making, it could be seen as a pro and a con. We have to be fast and efficient. So there is no time to really spend much of the time to perfectly control things. You really have to be smart from the beginning on, and we call this design of experiment, DOE, and quality by design. So we really invest in preparing for some methods to be ready, and then being fast, because time is money. This is of course true, and so therefore we have really to be smart and do what is needed, requested, and being really high quality delivering products in the end. Time to shine, I mentioned in the beginning. This is what I mean, that their own ideas are really implemented. You can have new ideas, especially when you have a broad background from academics. You might know techniques which people here are not aware, and which might lead very easy and fast to some measurements, which is better than identifying other excipients of products. So this is what I really like from the job, and also Harald area. I think it was also in the video. You really have a good work-life balance here. Just seeing this beautiful picture, I always get to rest just by seeing it, and this kind of stands in a good relationship to the stressy work environment, what you certainly have. So with that, I want to maybe just add one more thing, because this is when I kind of recap this presentation, I thought, okay, what is now the difference between industry and my let's say how I see industry before I entered the job, and how the reality is. So I was thinking industry has tight timelines. It is a money-driven decision world, and you have a high workload. So this is what I naively thought would be. And the reality actually is that, yes, there are tight timelines, but this is actually good, because this drives the projects. It's not money driven, actually it's quality driven, but it's money controlled, because of course we have to care of patient safety quality wise, but we also have to make some money to invest in the future. So money also plays a certain role. So sometimes projects are canceled because just business case is not good. But then we don't start the projects even. We decided that earlier. And the high workload is also true, but here, as I said, keeping the balance, you work hard and you rest hard. So it's really appreciated and accepted here that you stick to your hours. When you're working efficiently, you have, I've never worked on a weekend, what was very new to me coming from academia. So you can do the job in 5 days if you are efficient, and then you really have 2 days to rest. And I think this makes also here the work very, very attractable to people. And I will just close this now with a quote by Albert Einstein. I think this is also important. If you never made a mistake, you have never tried anything new. And so we are really always curious. We try to interact, we try to evaluate new things, new ways of thinking, reimagine medicine, what Cornelia said in the beginning is really highly appreciated here. So if you're a curiosity-driven scientist with a broad background, I think you will find your niche here, and you will get a good career path in the future by joining Novartis Sandoz. And with that, I want to thank you, what is standing on the last slide of my presentation, and I'm happy to take any question you have.
Thank you very much for your talk. If there are questions, we're now going to move on to the Q&A. And if you have questions, we highly encourage you to just raise your hands in Zoom and unmute yourself. Turn on your video camera and ask questions. If you are really shy, you can also put them into the chat and I will read them out loud for you. There's a hand. Okay. So thank you. We're going to move on to the Q&A now. Renata.
Hello, Harald, and thank you for the talk. I wanted to ask you if somebody that just finishes their PhD in basic science would be interested in looking for a job in Novartis, what would it be enough just to have a PhD? For what type of jobs would there be available for this kind of people?
Yeah, hello, Renata. Thanks for the question. Actually, you necessarily do not need a PhD. This is something what comes on top of it maybe, but of course with 4 years of PhD you might lose some time what you can spend in getting trained at the university, but in every level we are searching for people. So there are positions like I have where you really own your own projects. For that, you might need experience, you might need a PhD, but also engineers are very welcome. So you just need experience, I would say. Yeah. The other thing is some bureaucracy, what comes with it. But we also have senior scientists here, where you have a master's degree basically, and then you start working below under the umbrella of a more experienced scientist. And by time, let's say 1-2 years, just to say some house numbers, it depends how steep your career evolvedness improvement is. You get promoted by chance. Yeah, if there is a free position and you kind of showed that you can work and that you are smart and drive projects, then there are many ways for you to evolve here, not only in the chemistry field, but if you see that this is maybe the wrong way to go, we have the biologics in Schaftenau where you can go protein expression and other stuff. Still coupled to analytics, so it really depends. So actually, if I wouldn't feel that much appreciated and happy here, I would have searched for jobs inside Novartis already yet. The earlier you find your niche, the better it is for the future. Yeah, to feel comfortable and being really in the job you want to be.
Okay, thanks. And I don't know if you could answer me this question, but what would be something on one person's CV who applies for a job that would be maybe interesting for Novartis, like knowing some specific techniques, like you mentioned, or something else?
Yeah, maybe a very recent issue. What we discovered here is that we have some lipid mixtures in some drug products here. Yeah, so you use them as an excipient, and for that, we were not sure how to quantify them in a good way. And then I said, well, I have 12 years experience in lipidomics. If I can measure 250 lipids in one single HPLC run, I might be capable of measuring 2 or 3 of them. And at the side, this new method also brings the beautiness that you can also detect impurities in such things, what you might not see when you do a QNMR on it. Yeah. So such things are highly appreciated. So whatever you have, I think you will find a way to use it here. You just have to talk to the right people. But in development, it's really interconnecting and not you have a team and you stick to the team, but you reach out to the experts, and that makes this beautiness what I got to know in academia and what also fills out here my daily life.
Okay, thank you.
Welcome.
So yeah, thanks for the talk. And it seems that you put a lot of weight on experience. So I guess my question is, if the experience is not specifically pharma based, but coming from food or other kind of, but with a lot of experience in method development and these kind of things, chromatography, whatever, is that maybe not a plus, but is it a requirement? How would you call it? Yeah, I don't know if that makes sense.
I haven't seen this as a requirement. I mean, you always see that experience in a job is kind of mandatory in many of applications everywhere. But when you fulfill 80% of some application bullet points, you might apply to that job. You don't have to be 100% match. If you bring the correct mindset, this is how I would phrase it. If you are curious, if you are smart, if you have this experience to be curious and smart in the same time, this is of course a big plus because this speeds up processes. If you go the right way without testing 17 different parameters, but you know this will not work, I know this. Let's go for the extreme of first, test how discriminatory a method is, then narrow down to a window where we go and optimize it afterwards. So if you can speed up this by bringing some experience, or as I call it, gut feeling, that of course is a plus, but it's not a must, because for such things I am here, or colleagues of mine, to really bring you into this mindset to work in this team, to interact, to ask the right questions at the right time, not be shy, try to shine and be really a part of the team. Yeah, I hope this answers the question. So the mindset is, I think, a plus, but if it's not there, there are ways to learn how we operate here in Novartis Sandoz.
Thank you. And I guess maybe the question is not specifically for you, but and do you know about for third-party nationals? Like, is that a hindrance, or?
I might give this question to Cornelia. Yes.
Okay. Yeah. Okay. Yeah. Thanks.
Happy to answer that question. So thank you very much. And this, I would have anyhow at the very end of the presentation said a few words, but happy to address it right now. So we, as stated on one of the slides, we are very, very open to all kind of nationalities. We have 76 nationalities already, and this would already give you maybe a hint that we are very diverse and we are very open to diversity. So when it comes to application, regardless where you're from, we support, if you are the selected candidate, regardless of your nationality, we fully support a relocation process, and if this requires an immigration process because you have a different nationality, we have a so-called really a global mobility team that works with the final candidate to support this person throughout the entire onboarding process, so helping with the entire, legal immigration documentation things that you need to prepare to get the working permit in Austria, but also helping you to settle either yourself or maybe even with family, if this is a situation, to help settle, find an apartment, clarify some taxation topics that you might have or something like that. So there's a huge support which we offer for every candidate that is successful in the application, and provide as much as possible the help to get started and onboarding.
Thanks a lot.
Questions in the chat. I will have a personal question. It is that you have said that you have worked quite a lot of time in academia, and you have done a postdoc, and I would like to know what if you had some type of culture shock in your daily routine when you changed from academia to industry, maybe positive or negative?
Very good question. I have to kind of fill some thoughts by talking about something else and maybe repeating the question. So when I come from academia here, the first shock what I had, openly speaking, is that there were a tremendously amount of meetings. So I mean, this was also Corona-driven, I think, of course, that you don't have live meetings. Normally in academia, you sit together with the whole group and discuss the things. Here, everybody was sitting in the office, having the headset on like we have now, or people are in home office and just contributing from at home. So this was a little bit of a shock, but this, of course, then turned out to be not an issue anymore when this big wave of Corona was over and we were allowed to work in the lab again very freely. Still, there are some meetings what could have also been solved by email, but this is I think also true for academia, that sometimes the boss wants to have something what you say, well, let's do this later, it's not on my priority list. So actually no, but talking about all this, there was no big shock. It was more, I was positively surprised about the efficiency. So that you are really fast, that you don't have time to control each and every single step in a subsequent way, but you have really to be smart to cover everything as soon as possible. Yeah. So the critical quality attributes of a material, for example, that this is defined before you start an experiment. So it positively surprised how efficient you can be if you spend maybe a week in the design of an experiment and then executing it, rather than trying to execute and learn by trial and error. What of course then comes afterwards, but in a more controlled way, even because you spent this initial phase in really designing an experiment and already interchanging with other people to get opinions, to evaluate and to optimize. Yeah.
Okay, thank you very much.